So I'll start then by actually looking at individual neurons before we finally look at networks. And if you haven't seen lots of different neurons before, this picture illustrates that actually neurons come in all different shapes and sizes. And just looking at a picture like this, you should kind of immediately wonder, why? Do all these different shapes actually matter? Are they just a question of enabling neural networks to form in an efficient way, or do these different neuronal shapes actually influence the way a neuron integrates its inputs from other neurons and produces an output? And these questions are actually rather difficult to fully address experimentally, but modeling, as in creating a mathematical model of a neuron and then simulating it on a computer, can really help address these sorts of questions. And this has become really the heartland of computational neuroscience, being able to model and simulate neurons in some considerable detail. And that's what I'll concentrate on first. So why model a neuron? I kind of already said a little bit about that, but basically experimental techniques still are rather limited in the control they can give the experimenter over inputs to a neuron and the recordings they can make of outputs. So typically it's only possible to stimulate and record from a few, maybe only 2 or 3 at most, points in a neuron. And if you're trying to control, say, synaptic input from other neurons to the neuron you're recording from, that is very difficult to do. It's experimentally, you can grossly stimulate a lot of inputs, but you have no fine control over the number of inputs and where those inputs are on a cell. But if we develop a model and a computer simulation, then we can actually have that fine sort of control over those aspects. So we can investigate the input-output aspects of a neuron in much more detail. So I'll just try and show you one little demo, which is one of the exercises you can play with this afternoon or later on. So this is a simulation built using Neuron. You see lots of little windows and pictures of the cell. I'm just going to run this now. Hopefully it'll do what I want it to do. So what we're seeing is the membrane potential on the right-hand side, 3 recordings from the 3 red dots here. The top one is the cell body. The middle one is a dendrite in the middle of the apical tree, and then one further away down the bottom. And you can see we can record the electrical activity in terms of the membrane potential at these different spots, which is something you can just about do, or you can do experimentally. But what we couldn't do experimentally is the stimulus that's been supplied in the simulation is actually an action potential going to 400 different synapses in these red dots here in the middle of the neuron, distributed over this dendritic tree. In this simulation, I can control precisely the number of those inputs that are stimulated. And in this example, I'll just show 400. But you can see also from the response of the neuron, it's quite complicated. So there are a couple of spikes here, these sharp peaks, which are known as action potentials coming out of the cell. But there's a longer, slower peak as well. And we'll look at that a little bit later on and use a simulation to give us some clues about what might be generating that. I'll just run this again. If you look at this shape window in the middle, as Gauta illustrated, another nice thing that we can do with simulations is to visualize what's going on in different ways. So we can use a color scale to actually also illustrate the membrane potential over the entire neuron while we're simulating it. So there are many good reasons to actually model and simulate detailed models of single neurons. And what I want to do now is just give you some idea of the components of such simulations. Basically, what we're doing is building up the picture of a neuron as an electrical device. And so we can develop a description of a neuron in terms of electrical circuits, as shown here. And what's underpinning that simulation that we've just seen is the concept of compartmental modelling. What we do here is to take the morphology or shape of a neuron, like this grey tracing you can see in the background at the top there, and subdivide it into small pieces or compartments, which are the blue cylinders shown in this schematic in the middle here. And what we then do is describe the electrical properties of each of these little compartments as an electrical circuit. The assumption one fundamental assumption we make in doing this is that the membrane potential across the membrane in a compartment is the same throughout that compartment. Now, in general, the membrane potential will actually change continuously along the length of a neuron. So we are making an approximation here. And so the number of compartments you choose will have a fundamental bearing on the accuracy of the results that you get out of it. Clearly, the more compartments you have, probably the better resolution you get, but at the expense of computational time. But that's one of the sort of key things you need to choose when you're building a compartmental model. But at the end of the day, you end up with a description of your neuron in terms of an electrical circuit. So every compartment is connected to its neighboring compartments via an intracellular resistance. And so we end up with this one complete circuit, which describes the cell. Now, I'm not going to go into details of how this actually works, but we're not dealing with electrons here. So the electrical currents are a result of charged ions flowing across the membrane. And the ability of different ions to flow across the membrane depends on the opening and closing of proteins which span the membrane and are known as ion channels. They can form holes in the membrane which allow particular types of ion, such as sodium and potassium, to flow across that membrane and create an electrical current. Now, if you don't know anything about this aspect of neurons and the electrical flow, or the flow of ions creating electrical currents, strongly suggest that you do get some familiarity with this concept, even if you're not really studying the nervous system at this level of detail, because it gives you a really basic idea of how neurons are actually working as electrical devices. So one thing I haven't said so far is that we can, well, I've said we can describe a small patch of membrane or compartment by an electrical circuit. The basic components you're going to have there are capacitance. So the membrane itself forms a insulation between the inside and the outside of a neuron, and that has the properties of capacitance in electrical terms. The membrane also has a fairly high resistance, which we model by resistance here, Rm, and there can be some flow of charge across the membrane itself, and that is powered by a battery, which we call EM. And so we'll see in a moment that different types of ion channels will represent also by a resistance and a battery to form a set of series circuits that connect together to give the whole picture of a single compartment. We might also have an experimentalist injecting current into our compartment too, and that's shown here as a current source on the right-hand side. Now, what we can do with electrical circuit description is turn it into mathematics. So Gout has already shown you some equations, but the basic fundamental equation that represents this circuit is that we can describe the rate of change of the membrane potential in this circuit, which is from the inside to the outside, as a differential equation. So we have dV by dt as a function of capacitance and resistance and the battery and injected current. Further still, there are numerical methods for turning that into a set of equations which we can code up in any computer language and create simulations such as the ones that I've shown. So that's just a single compartment. When we join compartments together to get a more complicated model, that just means we have lots of these equations. So now we subscript them with, say, J for compartment J. And so that might be the middle compartment here. We have J plus 1 and J minus 1 compartments, which we join together as well by an axial resistance RA. And we can write down the equation again for this entire circuit. And we can keep adding to that as we have more compartments. And you can end up with hundreds, if not thousands, of differential equations describing your neuron, which we can then still turn into numerical mathematics and code it up so we can run simulations. So one of the basic things that most of these simulations will include is some mechanisms for creating action potentials. So the action potential is the fundamental signal that goes between neurons and is in the form of a rapid change in the membrane potential. The amplitude is about 100 millivolts and it lasts, as you can see here, for 1 to 2 milliseconds. And it's generated by the flow of sodium ions, firstly sodium flowing into a part of the neuron, typically the start or the axon, which depolarizes the neuron. So the resting voltage is a negative potential around, say, minus 60, and as sodium flows in, the membrane potential goes upwards. And then later on, potassium starts to flow out of the cell. The sodium and potassium are both positive ions, and that will actually cause the membrane potential to drop back again, creating this voltage pulse. Now, one of the most famous and most important results in computational neuroscience is this phenomenological model, essentially developed by Hodgkin and Huxley and colleagues back in the 1940s and 1950s. And they actually had to use a hand calculator to calculate this fundamental equation to solve a model that simulates action potentials. So they developed fundamental components which describe the opening and closing of sodium and potassium ion channels and how that changes over time. So as you can see with this description here, initially I've just had sort of static resistances. We're using G now for conductance, which is the inverse of resistance. And the sodium and potassium conductances do change over time, so hence these arrows. And it's these changes in conductance which cause this action potential. So Hodgkin and Huxley came up with a mathematical description of the dynamics of these conductances, which form the basis still of the way that we model a variety of ion channels. So though theirs was a purely phenomenological description, actually what they came up with in terms of the dynamics of ion channels depending on what they called gating or so-called gating particles, N shown here, actually turns out to be a pretty reasonable description of how the proteins that form ion channels actually work. You can get, and so the types of equations they use are actually still the sort of equations that we use in developing ion channel models today. So again, I'm not, as I said at the outset, I'm not going to look at the details here. If you don't know the Hodgkin and Huxley model of action potential generation, again, I really urge you to study that. And it's certainly in pretty much any good neuroscience textbook will cover this model, at least, if no other models. And I've given the full details here, which you can also study if you like, but I'm not going to talk about it anymore in detail. But what we can do, given a model of action potential generation, is also develop models and simulations which are very hard to look at, situations which are very hard to do or treat experimentally. And so one thing is to look at action potential propagation along axons. So just you should hopefully see some pretty colors on the left-hand side there. So what you can see here is actually a reconstruction as a compartmental model of a section of CAT, motor neuron axon. And you can see it, so it starts here and goes along. There are branches in the axon, and there are swellings known as boutons, where actually typically it forms synapses with receiving neurons. But what we can do, which you cannot do experimentally still, is actually, I'll just replay that one, visualize and look at the flow or the spread of the action potential along this sort of complicated axon. And even if you study this closely, you can see that, whoops, there's the next one. We'll come back to that actually. The first one, you can see there are time delays involved in this. So the action potential won't actually reach the end of an axon, each of these terminals, at the same time. So that's maybe something that's significant in the nervous system, whether these time delays in transmission actually matter in terms of network properties. Something else we can do experimentally is actually change the properties of our axon and study this sort of phenomena. So all these branch points will most often cause some sort of increase in the electrical load on the signal flowing along the axon. So getting through branch points can actually be a little bit tricky. And what we've done in this second simulation is simply to reduce the maximum conductance of sodium channels throughout this axon. And as soon as we do that, we get the action potential flowing through most of the axon, but you can see it fails at one of these branch points. Nearly gets through, but doesn't quite make it. So that's a really useful, or two useful things we can do with these simulations, is study the speed of transmission along axons and also the safety factors involved in terms of how reliable transmission might be. And these are things that are not really possible still to study experimentally. So there are lots of ion channels now known about in terms of the nervous system. So experimental data, there is a wealth of data about the different ion channels. So these are things that can be well studied experimentally. And so the characteristics of many different ion channels are well known and can be used in simulations. What we still don't know, though, and is still hard to gain experimentally, is the exact distribution of different types of ion channels, both across the brain, but also within individual neurons still. So we do know that many neurons will have quite a number of different ion channels in their membranes, and will possibly have some crude picture of how they're distributed in the membranes, but not anything with great precision. And typically still, different types of ion channels will not be fully characterized in all types of neurons in which they appear either. So Gout has already mentioned the difficulty of getting parameters for simulations. We are still in the game of actually having to use parameter values which have not necessarily been determined in the neuron that we're actually modeling. So Some approximation and guesswork is involved, and a lot of optimization can be done in terms of tuning a model to match particular experimental results by changing parameters. So I've only mentioned really sodium and potassium ion channels as underpinning the action potential. But one thing we're using modeling to do extensively is to try and understand the functional role of different types of ion channels. And one of the exercises we can play with this afternoon actually uses a different type of ion channel, potassium ion channel, than the one that underpins the action potential. So the potassium current underpinning the action potential is a delayed rectifier current. There's another one which is common in many neurons known as the A current, which has different dynamics and it can do different things. So again, I'm not going to explain the details here, just to highlight that we can play with a simulation later on that will explore the role of the A current in neuron input-output. And what we'll discover is that actually this potassium ion current might be or can be one of the mechanisms which determine whether a neuron will fire with type 1 characteristics or with type 2 characteristics. And I will just try and explain this briefly, what these mean, though we can talk about it more this afternoon by running another little simulation as well. So let me just fire up another neuron simulation. Right. Let's run this quickly. So what I'm doing here, we've got a very simple neuron simulated behind the scenes here. So you can see it's actually just a soma or cell body with a single cylinder representing the dendrites coming off that. So it's a relatively simple model, and we're stimulating it in much the same way that an experimentalist can most typically stimulate a neuron, by injecting current into the cell body. At the moment, there's no current, so the cell is just sitting at its resting membrane potential. I'll add a small amount of current to it, and you can see there's a little blip, but then it returns back to rest. So this injected current, which lasts throughout here, actually is causing some of the sodium ion channels to open and generate a small electrical current. But they tend to close again, which is known as inactivation after a short period, and the membrane potential drops down. But we're not getting an action potential yet. Up the amplitude a little bit more, we finally reach a membrane potential, which is the threshold for action potential generation. So as the membrane potential goes up, more and more sodium channels will open until eventually they cause this regenerative spike, the action potential. But in this cell, we only get the one at this amplitude and it comes back to rest again. Now, what you can do with, or what is done experimentally, is to determine the so-called FI curve of the neuron, which is the frequency of action potential output versus the magnitude of the current you're injecting. So in this case, there's a startup, which is generally gives you one action potential, which we kind of discount in the FI curve, and this comes back to rest. Now I'll just up the amplitude a little bit more, and now we get a nice stream of action potentials coming out. So if we do that for lots of amplitudes and plot the frequency of these spikes or action potentials over time, we get an FI curve. And what we've just seen is the Hodgkin-Huxley model of action potential generation without anything else happening in the neuron gives you type 2 firing. This means that for small currents being injected, you don't get any action potentials, or maybe that one initial one. But then at some point, a small increase in the injected current will suddenly cause a non-zero frequency of firing. So the neuron will start to fire continuously at some non-zero frequency, which then might increase a bit within further injected current. What you might think is a more interesting and useful FI curve might look like the one shown just above that, which is known as type 1 firing, where initially the cell is silent, but you get to a certain amplitude where now you get a more or less continuous, almost linear increase in the firing frequency with injected current, which I guess mathematically is a nice sort of transfer function to have. That's known as type 1 firing, but is actually rather hard to achieve in neurons. And the A-type potassium current is one mechanism that can cause type 1 firing by essentially slowing down action potential generation as the threshold is breached. So it basically drags this high firing frequency down to lower levels for those sorts of injected currents. And we can use simulations to discover whether that works or not, and we can play with that this afternoon in the tutorial sessions. So that was done with a very simple neuron model. I started out, though, showing you an example of a rather more complicated compartmental model of actually what is a hippocampal pyramidal cell. So the black lines you can see buried under all my pretty colored dots here is a tracing from a real hippocampal cell. So experimentally, we can now actually stain and visualize neurons in the entirety and digitize their morphology, which is a really nice tool then to be able to form complicated or detailed compartmental models of such neurons. And this particular simulation I've shown you involves dividing this cell up into 337 compartments and distributing 6 different types of ion channels across those compartments. So one important thing about these sorts of neurons is that the distribution of ion channel types is very typically heterogeneous, by which I mean there are distinct gradients of ion channels across these membranes, across the dendrites. So the electrical properties of a neuron can be quite dissimilar in different parts of the cell, leading potentially to highly nonlinear signal integration and processing within the dendrites themselves, leading to the final generation of output from a neuron. And simulations can really help us to understand that sort of signal processing in these cells. Just to get back to the initial simulation I showed, one thing we again can do experimentally, which we can't really do as cleanly as we'd like, do with simulations we can't do well experimentally, is to actually control the distribution of our ion channels in the membrane across the cell and the density or maximum conductance of those ion channels as well. So one thing I can nicely do in this cell is actually simply remove or reduce the conductance of a particular type of ion channel. And so what I'm going to do now is remove what's known as an R-type calcium channel from this cell and rerun the simulation. And you can see that apparently there was a calcium current which was contributing a lot to that second action potential spike we saw in the initial simulations. So that's kind of a simple example which shows you the sorts of experiments we can do with our simulations to try and understand how what contributions different ion channels make to these cells. And so yeah, it's just illustrating what you've already seen in the simulation. Okay, so that I spent a lot of time on this, but that kind of introduces you to one of the fundamental building blocks in computational neuroscience, which is compartmental modeling of neurons, where we build up a fairly detailed description of the morphology of a neuron and the electrical properties across that neuron in terms of ion channels and their electrical properties. Now, there are so we're building slowly to getting towards networks of neurons, but there are other things you might want to do with single cell models as well. So there are good reasons for why actually you might want simple models rather than these complex models. And there are different approaches to actually achieving simple neuron models. So just this picture illustrates here, one thing you might consider is looking at a complex cell like this, that actually it's, apart from the fine branching details, the basic structure of the cell is really quite straightforward, in that you have a cell body down here somewhere, and then you have a lot of dendrites going upwards, which are the apical dendrites, and a lot of dendrites going downwards, known as the basal dendrites. So you would think, and it's true, you can capture the essence of this morphology in a much simpler structure, using a few compartments to capture the main morphological features of a neuron. And that is something that you could certainly do. And the two main reasons why you might want to do this, well, A, it's computationally much simpler, so it's quicker to simulate these simple neuron models. Though, as we're seeing, computer power is getting to the point now that actually we can simulate complex neurons quite readily, even on our home computers. So I'm just running on a laptop at the moment and doing that single cell model quite easily. So computationally, there's still good reason for doing or reducing our model to a simpler state as possible, particularly if we want to build large networks of such neurons. But also theoretically, a simple model is typically much easier to understand than a complex model like this. And so if we want to get at the heart of the properties in terms of signal processing of a neuron, generating as simple a model as possible that captures the signal processing capacity of a neuron will probably aid our understanding greatly. So that's the other main reason why we might want to reduce our models. And I'm just going to skim through some of the approaches that have been taken to reducing single cell models before finally getting on to networks. So I've just illustrated that we can get what's known as bursting behavior in a compartmental model of that hippocampal neuron, underpinned by a calcium current, where single volley of input can create a little burst of 2 or maybe more action potentials coming out of the neuron. Now we can get the same behavior actually by reducing our model down to just 2 compartments. One compartment being a soma, the other compartment being a dendrite, where the electrical properties of the 2 compartments are different. So our dendrite contains calcium channels, which are absent from the somatic compartment. And this was done back in 1994 by Pinsky and Rinzel to create this simple reduced model, which has quite complicated firing properties in terms of what it produces for a given magnitude of driving current coming in. So it's a really nice approximation to that complicated model and gives you great insight and control over bursting mechanism in such a cell. There are other approaches too to creating simple spiking neuron models. One approach does still generate action potentials like the Hodgkin-Huxley model, but reduces the Hodgkin-Huxley model as much as possible. So mathematically, there are different ways you can actually reduce the number of state variables down from 4 in the Hodgkin-Huxley model down to 2 to create a model that still generates realistic looking action potentials. And there are a variety of approaches to that, as I've listed here. Now, a further simplification that we might want to make, which is particularly important for creating large networks of neurons, is to actually throw away simulating the action potential itself altogether. So the action potential, we can somewhat assume, is just a standard signal. Its characteristics don't change much. And what we're really interested in is when an action potential is generated as an output, not the actual form or shape of that action potential. So one approach to doing this then is to create what's known as an integrate and fire model, where we model the subthreshold membrane potential in some detail, but then track when that membrane potential reaches some predefined threshold, at which point we say, or the model says, that it's an action potential has been fired. So the only output now is the time of such an action potential, not the actual waveform itself. So this greatly simplifies the numerics involved or the calculations involved, because we're not actually modeling the ion channels underpinning this. So the sodium and potassium ion channels are not being modeled. We don't have to model the time course of the action potential, just the sub-threshold changes as a result of inputs from, say, an electrode or from other neurons. So integrate and fire models are widely used still to create networks of spiking neurons. Not only are they cheap to simulate, but actually they're simple enough that you can do some good theoretical analysis on the properties of the networks that you might be simulating and the responses of individual neurons. So there are various flavors of integrate and fire neuron model out there, which you'll come across, which add more or less complexity to the basic model to try and make them slightly more realistic in terms of action potential firing over time and the timing of that. So you might need to add another equation with another variable to get adaptation in spike timing, which is something that's actually common in many neurons. And exactly the characteristics of how the threshold is reached for action potential firing has resulted in development of some more complex approaches to the sub-threshold membrane potential as well in integrate and fire models. But they're still much simpler than a detailed compartmental model. So this will lead us in this talk eventually to looking at networks, which we'll get to in a moment. But to do that, of course, we have to connect our neurons together. So I've already been using a model of synaptic input, which is the signal which is generated on a receiving neuron, the postsynaptic response to a presynaptic action potential, or an action potential coming from an input neuron which synapses on the postsynaptic neuron. So the hippocampal model I've shown has been modeling that process. Now this schematic here shows that actually the synaptic transmission process is hugely complicated. It's a very dynamic process, and the details here are something that also is the subject of significant computational modeling to try and unravel all the details. I'm not going to talk about that today, though it's something I have been involved in personally over the years in modeling the details of synaptic transmission. But I'm going to introduce the most basic approach, where actually all we want to do is to model the postsynaptic current that results from the arrival of an action potential at the synapse. And there are three basic approaches which are mathematically very simple. So all we do is phenomenologically model the waveform of the postsynaptic conductance that results from an action potential. And three commonly used equations are shown here. The most flexible is the, but also most computationally expensive is the dual exponential, which gives you control over the rise time and the fall time of your waveform. Then the resulting postsynaptic current is the product of this conductance with the difference between the membrane potential at the synapse and some synaptic reversal potential, because the synaptic current is also the result of ions flowing across the membrane. So there is a battery with a reversal potential involved with that. So given such simple models of synaptic responses, we can then create networks of spiking neurons, connect them together, and investigate their properties. And I'm just going to give you a couple of simple examples, again, which you can play with in the tutorial this afternoon, of the sorts of questions that you can ask and try and answer by doing such simulations. So here's one that actually it's a really interesting question that's been around for a long time and was for a while, particularly back in the 90s, a subject of considerable debate, and probably still is actually. And the issue is this, that actually recordings of spiking from many neurons, such as the little spike train shown here, indicate that actually spike firing is often very irregular. Now, this could be due to simply the intrinsic properties of the neuron itself, or it could be a property of the inputs it's receiving. Now, neurons are in fact noisy devices. Ion channel opening and closing is a random process, but that level of noise is typically not sufficient to account for all the irregularity in the spike firing we get. So it's more likely that there's a significant contribution to the inputs that the neuron receives. And so there's been an awful lot of work running computer simulations of spiking neurons that show that actually you can get this irregularity, but only with a very suitable balance of excitatory inputs and inhibitory inputs. So I should explain that an input from a neuron is excitatory if the postsynaptic current results drives the membrane potential upwards towards the action potential threshold. That depends on the reversal potential here. So excitatory synapses tend to have a reversal potential of around 0 millivolts. Alternatively, other inputs to neurons are inhibitory in that the postsynaptic current that they generate tends to drive the membrane potential down towards the resting potential rather than the firing potential. So they're known as inhibitory inputs. And input neurons will either be excitatory or inhibitory. And so, as I was doing with that hippocampal pyramidal cell, we can play or generate inputs to a neuron that are both either excitatory and inhibitory, and investigate the spiking output that results. And we can demonstrate with a simulation that it's only very exact balance between excitation and inhibition that results in this irregularity. And I'll just quickly run this simulation. So here we have a suitable balance. So what we're actually simulating is just a single integrate and fire neuron, which is receiving lots of inputs. In this case, 300 excitatory inputs and 150 inhibitory inputs, where the inputs themselves are random spike trains from each neuron. And this plot then is mostly the sub-threshold membrane potential, so the noisy train here, and these square bits just show when an action potential has been signaled and fired from this cell. And if we do this for long enough and calculate the interspike intervals between each of these spikes and generate a histogram, you end up with this sort of histogram, which actually indicates, or is rather an exponential distribution, so a random distribution of spike times coming out. But then if we reduce this to just getting excitatory inputs, which are still random in themselves, you end up with a much more regular interspike interval from this. And this is because the neuron itself has a lot of filtering aspects to it, which cause it to more or less want to fire fairly regularly. So even if it's getting random input of a particular nature, just excitatory, it'll fire. It's more likely to fire regularly than not. And so you actually have to really tweak it by giving it suitable inhibitory input at the same time to get it to fire randomly. So of course you can investigate this too with networks of neurons, and we have a simulation you can play with that does that as well. We've now got lots of inhibitory neurons and excitatory neurons which are connected together in different proportions, and you can also create random firing in these neurons. But you'll also discover in the tutorial, if you play with it, that it's quite sensitive to the balance between excitation and inhibition. And even where you get lots of random firing like this, you might get periods where the cells start to synchronize and fire more regularly for a while before retreating back to random firing again. So it's just rather one simple question that simulations can provide a lot of insight to. The alternative to such random firing, though, is regular firing, and the brain does exhibit a lot of regularity in its firing. So there are a lot of different rhythms that we record in brain activity, and exploring those sort of rhythms and how they're generated, how different frequencies of rhythm are generated, that is also the subject of a lot of modeling and computer simulation work. And I'm running out of time now, but I will just say that there is a simulation we can run this afternoon, which also uses excitatory inputs, neurons connected to inhibitory neurons, but now they act as oscillators rather than as creating random activity. And the EI oscillator is a fundamental component of many rhythm generators. So I'm just about done. The remaining part of this talk pertains to other simulations in the exercises, which we won't have time to look at today, but you might look at later on, but also covers topics which you'll hear in great detail about over the next couple of days, which is to do with the obviously very interesting topic of learning. So one of the main points of us, I suppose, is as creatures, our brains are able to learn about the world around us and change our behavior as a result. And the basic component of that learning, we think, is the concept that the strengths of synapses between neurons change according to so-called learning rules to enable us to learn and recall information. And so one of the simulations I don't have time to talk about now that you can play with is a model of a network of neural network acting as an associative memory, where we can store and recall patterns of information by changing synaptic strengths. But as I say, you'll hear a lot more about learning rules, plasticity in the nervous system, but also the function of neural networks as a whole, such as associative learning, over the next few days. So there are lots of details in the slides that you can look at for yourself later on. But at that point, I'm actually going to call a halt to my talk and just see if there are any questions. As I say, in quite a short period of time, we'll get getting back and looking at the tutorial exercises. And before that, Arne Roth is going to tell you a bit more about the Neuron simulator and show what it can do in some more detail as well. So at that point, I'll stop and just stop sharing my screen and see if there are any questions.